these mice do not have repetitive problems with repetitive behavior. So right here we have isolated a mouse model that has 2 of the phenotypes, but not a 3rd phenotype that belongs to the domains that are used to diagnose autistic kids. Really quick, Cereblon was a gene, another mouse model we were working with, that Joe Higgins identified in a family that lives in upstate New York, and he identified this gene, Cereblon, that has a point mutation, very kind of similar to what Francis talked about. And these kids have intellectual disability and mild autistic phenotype, and we have gone ahead and made the knock-in mouse now, and we're characterizing it similar to what I just described. And really quick, in the last couple of slides, as I said, in the end, we are interested in using our mouse models for drug discovery in the hope that we can help kids. And even though it's a long journey, it's a great start to have some of these mouse models to begin exploring. So I have to say that I'm extremely fortunate that the Hartwell Foundation decided to fund a collaborative project between a colleague, Andrew Pieper, and myself to test and use a very novel strategy that Andrew developed a few years ago. And in this strategy, what he does is he said, why not test compounds, novel compounds, directly in the mouse? So what he does is he has been using a compound library. So this is a pool of novel compounds that we don't know what function they have in the brain or in behavior or in a living animal. He screens those compounds by directly putting them into the brain of mice. And the whole idea is that to identify molecules that in our mice, which is the project that we have ongoing, is to try to identify novel molecules that would be able to rescue the behavioral phenotype, for example, in the calcium channel knockout mice that I described. So he's currently following the strategy. The mice have been implanted with the different compounds, and he's testing whether any of them will alleviate any of the deficits that we have seen in these mice. One of the compounds that Andrew had already identified, it's called P7C3. A couple of years ago, he identified this molecule that rescues some of the learning and memory deficits in some of the disease mouse models he was studying. And the interesting thing about the series of compounds is that it also rescues in the brain a deficit called, so the brain has, there's a part of the brain called the hippocampus that is very important for learning and memory, has been implicated in anxiety, and also some of the social deficits seen in mice or in animals. He found that this series of compounds can rescue deficits in neurogenesis. What's extremely, I don't have the data here to show you, but what's very, very new data we have with our calcium channel knockout mice is they have 50% deficit in adult neurogenesis in the hippocampus. So they're not making these new neurons in the brain, and we're very curious why, which we are studying in the lab. But what Andrew is testing is whether P7C3 can rescue and bring back these new neurons, which may then help alleviate the behavior. So this is kind of an example of where we are going with the drug discovery. And just to wrap up, I just wanted to show this slide again is as a summary of how we are we are looking for novel genes that in a patient population compare them to the parent genetic profiles, and then use that data to generate mice, which we can then use for drug discovery and take back to the patient. I have to profusely thank all the families, and I'm calling them the WCARP families, but the families who have patiently participated in our study and are doing so every Wednesday. We really appreciate their their generosity of time and their kids. None of this work would have been possible without the generosity of the foundations that we have got funding for everything that I've talked to you about today. And there is no way I can, that I can tell you how much I appreciate and how thankful I am for the collaborations, for the support of collaborators, our clinical team, which is a very new avenue for me as a basic scientist here at Cornell. And some of them may be in the audience, but bringing together, it's extremely exciting for me to actually have bringing together the clinical side at New York Presbyterian and Weill Cornell to our basic lab, and for us to be talking to each other and thinking about the big picture questions, but still using basic animal studies to inform ourselves about autism. And I absolutely am amazed at the dedication that my lab members have to all the work that we've been doing in the lab. So thank you.